Today we have Associate Professor Brad Turner. He is a National Health and Medical Research Council Dementia Research Leader Fellowship Fellow, and the NHMRC, as we call it, is our national government funding body, so very important to Brad and the Florey. He's also the head of the MND laboratory at the Florey Institute. He obtained his PhD in biochemistry at the University of Melbourne. In 2005, he undertook his postdoctoral training in mouse functional genetics at the University of Oxford for 3 years before returning to the Florey, and he has established here his independent research group. Associate Professor Turner is an MND expert, that's why he's here to talk to you today, and he leads a large team of researchers and graduate students investigating the molecular basis of MND using patient stem cell derived and animal models to develop drug, gene and immune-based therapeutic approaches, which he will talk to about today. His team has recently embarked on a world-first drug screening program for MND, which he will talk about in today's lecture. So without further ado, Brad Turner. Thank you.
Great. Thank you, Jane, for the introduction. And what a great week to deliver this lecture in the lead up to the big freeze at the MCG. So this is the FOD-MND Neil Danaher's Foundation. And what I'll talk about today is how their support of our laboratory and institute has really transformed the way we're addressing treatments of motor neuron disease. So it shows you how support can have an impact. So before I begin, I just wanted to show you this slide. This is a slide generated from the laboratory. And what you can see here are these red cells, and these are the motor neurons. So these are the nerve cells affected by the disease. We have about 6,000 on each side of our brain, and you can see they're quite pretty looking, and they've got these long connections here. And so these are the cells that are lost and disrupted in the disease. So just an overview of today's lecture. I thought I'd give you a brief history of MND, motor neuron disease. It's also known as ALS, amyotrophic lateral sclerosis. And I thought today I would give a different perspective. I'd give you the Australian version of motor neuron disease and what impact Australia has had on this disease. I'll talk about the causes of MND, what we understand to be the genetic and environmental factors that are involved in the disease. It's quite a complicated disease. I'll mention briefly clinical trials, what's happening in the clinical trial space in motor neuron disease. And then, of course, I'll talk about our establishment of a high throughput drug screening facility at the Florey Institute. So let's begin with the history of MND, ALS. And I guess for Australia, it all started with this chap here. So this is Alfred Walter Campbell, and he's actually a very distinguished neurologist. He's actually considered the first clinical neurologist in Australia. And he was trained in Edinburgh and he practiced in the UK, and he returned to Australia in about 1905. And he published a very famous monograph, which is called Histological Studies on the Localization of the Cerebral Function. This is a document that was published in 1905, and what it contained was he essentially was able to map the architecture of the brain. So he did some very detailed structure and functional studies, and in collaboration with another prof called Sherrington, they were able to determine the region of the brain that controls motor function. And we've referred to this as the motor cortex, and this is a strip of the brain which contains those motor neurons affected in MND. And this part of the brain is involved in sensory input and converting that into voluntary movement. But he did some more studies, and what we can see here, this is a slice of brain tissue, and this is under the microscope, everything's drawn by hand, and he was able to sketch out these different nerve cells, and you can see that they vary in size and shape as you get deep into the brain tissue. But he was particularly interested in these enormous cells in this layer 5 and 6, and these cells he actually sketched out, and these are actually the motor neurons that reside in the brain, and they're also called BET cells. So these are some of his very early diagrams of these cells. But he went one step further, and that is he characterised a couple of cases of patients with motor neuron disease. And in this particular case, he did work in an asylum at the time. This male patient, who was about 35 years of age at the time of diagnosis, complained of symptoms, and these symptoms involved muscular pain or spasm, and also signs of weakness. And these are the two features of motor neuron disease in its onset. And he recognised that this was ALS or motor neuron disease, but he went a bit further and he noticed that the wasting had spread to the muscles of the body. So this is another characteristic feature of motor neuron disease, this progressive spread of weakness and paralysis. But what was clever about Campbell's study was he examined this patient at autopsy under the microscope, and he focused on these large BET cells in the motor cortex, and he counted them, and he noted that about 85% of the cells had been completely swept away. So he had actually defined the pathological hallmark of motor neuron disease, which is the loss of these giant cells. But furthermore, he noticed that the loss of these cells was unique in that cells occurring in other regions of the brain were intact and preserved. So this speaks to the selectivity of motor neuron disease for motor neurons. So what happened next was, I guess, the first clinical description of MND in Australia, following Campbell's original reports from the UK, were by two neurologists. So one was Housen, so he reported a case of what he called muscular atrophy in 1918, and this was followed by Murphy, who reported a case of motor neuron disease in 1924. So this was the beginning of the emergence of motor neuron disease diagnosis in Australia, and it was published in this famous journal, which is the Medical Journal of Australia. So the next step in this odyssey was, I guess, in the 1960s. There was an appearance of a disease. It was an endemic disease affecting the Aboriginal population of an island called Groot Island in East Arnhem Land, as shown here. And these Aboriginals were coming down with a syndrome which resembled motor neuron disease. And in fact, they called this Groot Island MND originally. And it was characterised by muscle weakness and wasting, but also stiffness of the body. And in fact, the Aboriginals referred to people affected by this disease as the so-called bird people, because they resembled water birds such as the heron, and this is an example of a wooden carving. And the disease was quite prevalent, so in every 1,000 people on this island or in this region, 6 people were impacted by this disease. So this was the first beginning of clusters of motor neuron disease syndromes in Australia. So I guess the next step was there was this worldwide hunt going on to solve the genes responsible for brain diseases. And Australia joined that front in the 1980s, about 1984, in which we knew of the existence of these large families affected by motor neuron disease. So we can see that the disease in this case is clearly inherited. So the individuals in black are affected by the disease. And it was actually the Australians who contributed families to this worldwide effort and clinical material and DNA. And you can clearly see the pattern of inheritance in this family. So we call this dominant, so it's the MNDs appearing in every generation of this family tree. And it was through this worldwide consortium and effort that the first MND gene was solved in 1993. And this is the original paper, and I've noted the two prominent geneticists and neurologists on this paper. So this was Australia's contribution. And this gene is called SOD1 or SOD1, and it was the first MND gene discovered. And this is a picture of the crystal structure, if you like, of the protein. So this is how it folds in nature, and it's actually dotted with mutations. And so this was the first gene discovered about 25 odd years ago. So this was a major milestone. So then something happened the following year, and that was there was a drug which was approved for motor neuron disease. So this is the first clinically approved drug, and it's called Riluzole. And this is it's also known as Rilutek, so it's taken in oral form. And this is the effect that it has on patients with motor neuron disease. So if you track these patients over time, looking at survival, we can see that patients taking Riluzole are afforded a rather small effects, protective effects. In fact, their survival can be increased or may be increased by 2 to 3 months. Now, the sad state of affairs is actually, this is still the state of play in motor neuron disease. This is the only approved therapeutic and has a very marginal effect, and we have not seen anything emerge in the last 25 years or so. So that will bring me to the rationale behind the drug screening program that we've established at the Florey Institute. So I guess not much happened in the motor neuron disease space for about 20 years until we had the MND Ice Bucket Challenge. And I always like to mention this, so you might remember this craze that swept the globe, in which people, from everyday people to celebrities, were dunking themselves with ice buckets. And here we have some famous faces. So this is Bill Gates, for those of you that can recognise him. This is Mark Zuckerberg. And even Trump got into the action there, although he got doused by some supermodels, so he had conditions. So this is actually a shot of the lab back in 2014, undertaking the challenge. It was quite a brave challenge at the time because it was the dead of winter, equivalent in the Southern Hemisphere. And this was a serious feat because it actually elevated the awareness of the disease and raised some serious funds. So globally, it raised about $200 million, and in Australia, about $3 million. And then I always like to remark, in that same year, we had more exposure for motor neuron disease. So we had the film called The Theory of Everything. And for those of you who haven't seen this movie, this is the story or the biography of the late Professor Stephen Hawking. And it was Eddie Redmayne's portrayal of Stephen Hawking that earned him the Best Actor Oscar. And in his speech, he dedicated his Oscar to everyone living with motor neuron disease. And here's a picture of Professor Hawking. He passed away recently. But then we had our own hero emerge, our own celebrity in 2014. And this, of course, was Neil Danaher. So most of you will be aware he's leading the Fight MND Foundation. He's leading a very public awareness campaign. And we've got the Big Freeze 4, which is occurring next week on the Queen's birthday. And he's brought a lot of exposure and awareness and funding to the Institute. And here we have Premier Andrews visiting the lab with Neil. And we've had many events. So we're close allies of Neil Danaher's Fight MND Association. So now this brings me to the facts. What are the facts of motor neuron disease? So what I can tell you is that this is a disease which impacts about 2,500 Australians, and we know that 2 people are diagnosed every day, and 2 people are lost every day to motor neuron disease. And this disease is different to other degenerative brain diseases you can think of, such as Alzheimer's and Parkinson's and Huntington's, which can linger on for decades. This disease is rapidly progressive and lethal, and the average survival of most patients is around 27 months from diagnosis. The lifetime risk of developing motor neuron disease is not trivial, so it's thought to be 1 in 300. So if you go to the cinema, that's 1 person in every cinema. And we know that the incidence of motor neuron disease is on the rise. So it's thought that in the last 10 years, the incidence of MND has doubled in Australia. This is a very costly disease to manage, and it was estimated that this is a disease which costs the Australian economy about $2.4 billion, and that works out to be about $1.1 million per patient. And there are many factors behind this, but the main one is this disease requires intense multidisciplinary care. So you need a neurologist, a physiotherapist, speech pathologist, dietitian, occupational therapist, and so on. So you need a cast of people managing these patients. So now I'm going to move on to the causes of motor neuron disease. So what do we understand to be some of the causes implicated? And I always like to start off with this pie chart. And what we do know is in that 10% of cases, there is an inheritance, and we call this familial motor neuron disease. And what this means is that there is a positive history of motor neuron disease in the family, so it has to be present in at least 2 of the generations of the family tree. But this is only true for about 10% of cases. So the vast majority of cases of motor neuron disease are of the sporadic form. And what this means is simply out of the blue. So if you consider Neil Danaher, he has 3 brothers. They all played professional football, but only he is affected by the disease. So the mystery here is what is the cause of sporadic motor neuron disease, which is out of the blue? Another way that we can illustrate this is if we use a pedigree or a family tree as shown here. So here we have a family affected by familial or inherited motor neuron disease, and you can see that the affected individuals are coloured in black, and this individual has passed on the gene to 2 of his children and subsequently 3 of the grandchildren. Where on the right side of the screen we can see sporadic MND. So here we have 10 individuals who are not related, seemingly not related, yet 3 of them have developed the disease. So there's no clear reason as to why this has happened. So let's start by talking about what are some of the possible risk factors implicated in the sporadic motor neuron disease. And I could probably reel off 100 of them. I've heard very different stories from patients and their families as to what they think what their theories are behind their disease. But some of them I'll mention today. So we do know that cigarette smoking has been associated with a 2-fold increase in motor neuron disease. We know that professional sports and athleticism is associated with an increased burden of motor neuron disease in some populations. Military service, so a lot of Gulf War veterans go on to develop motor neuron disease, and the Department of Defence in the States is very interested in why this is occurring. This is an illustration of a virus, so that there's some ideas that maybe viruses could be lurking within the brain and could be responsible for this disease. And then lastly, this little critter here is actually blue-green algae, which I'll talk about. So this is a microorganism. So let's just go through some of these. Let's start with sports. So we're talking here about elite professional athleticism and sports. And one of the most famous examples of someone who contracted motor neuron disease in their professional time was Lou Gehrig. So Lou Gehrig was a professional baseballer, and he was struck down with the disease after 17 seasons of playing baseball, and he had to retire. And he gave a very formidable speech. So Lou Gehrig's a good example. Neil Danaher is another. And I recall there was this study published about 10 years ago in which a group of people had followed a soccer team of Italian players over a decade, and what they had established, what these players had actually had about a 6.5-fold increased risk of developing motor neuron disease compared to the normal population. However, when you really drill into this study, you find that it's not as convincing. So they analysed about 7,500 players over 10 years, and they expected about 0.7 cases of motor neuron disease. And what they found was about 5. So in the end, it was only 5 cases discovered. And there are many other studies which dispute the link between professional sports and athleticism and motor neuron disease, so it remains controversial. So let's move on to viruses, and when I talk about viruses, I'm referring to a particular virus, which is called a retrovirus. So what is a retrovirus? They look cool. And this has been a really enduring idea in the field and for the last 10 years, and it was really this publication in a scientific journal a few years ago which suggested some evidence for a viral infection that could be linked to motor neuron disease. And what this group did was they were able to take autopsy material of spinal cord from patients who have died from motor neuron disease, and they detected the presence of viral particles inside these nerve cells, yet the control patients were completely clear. And this led them to propose that perhaps there could be a viral agent lurking within motor neuron disease. And the idea behind this is that about 9% of all your genes are actually viral. So throughout evolution, viruses have inserted themselves into our genes, and they lay dormant. And it's thought that the idea behind this is perhaps in motor neuron disease, these viral genes are activated and they assemble viral particles, and these viruses can then infect motor neurons and spread through the nervous system, causing damage. And this theory would certainly be consistent with the spreading nature of motor neuron disease throughout the body. And what's fantastic is, at the moment, the best way to address this hypothesis is to test an antiviral agent in motor neuron disease patients. And this is a trial which is being conducted through Neuroscience Trials Australia, embedded within the Florey Institute, and it's called the Lighthouse Trial. And what they're doing in this trial is they're taking a drug called Triumeq, which is a cocktail of antiviral drugs used for HIV patients, of which there must be hundreds of thousands of people on this drug, and they're testing the effect of this drug in motor neuron disease patients as we speak. So I think this is a really exciting direction. So now we're going to move on to blue-green algae. So this was the picture that I showed you before. So what is the link between blue-green algae and motor neuron disease? And I always like to tell this tale because it's a great tale of epidemiology and motor neuron disease. And this is a story which really started in the 1950s in the island of Guam, which I guess is always under threat by North Korea. And what was occurring on this island was there was a group of people called the Chamorro people, and they were suffering from a disease which looked very similar to motor neuron disease. And they referred to it as litigo-bodig, and this translates into paralysis and tremor. And here we can see a victim of this disease. So this man is holding his wife, and she has very pronounced wasting and paralysis. She can no longer walk. So this got medical scientists interested in what was causing this disease. And I'll just point out that this disease, this form of motor neuron disease, was 100 times more common than anywhere else in the world in the 1950s. And people began to suspect an environmental agent or a toxin, and they looked to this particular plant. And this plant is called a cycad tree, and it produces these edible seeds. And the locals had been eating these seeds for decades. And it was known that the seeds contained a toxin, and the locals were very aware of this. And what they would do is they would cover up these seeds and they would wash them repeatedly to rinse out the toxin. And then they would grind up the seeds to make their tortillas. So this got, the medical researchers were quite confused by this. They thought, well, if they were cleansing out the toxin, how was it actually entering these people? And then they noticed that the island was occupied by these fruit bats. And these fruit bats were voracious eaters, consumers of these cycad seeds. Yet the fruit bats were immune to the effects of the toxin. And then it was discovered that the Chamorro people love to dine on fruit bats. This is where the plot begins to thicken. They love fruit bats. They eat them for breakfast, lunch and dinner. And this was part of their diet. And so it was theorised that perhaps the fruit bats were the point of entry of this toxin into the Chamorro people. And with a bit more investigation and study, it was found that within the roots of this particular plant, there exists blue-green algae. And this makes this toxin, this poison, which then concentrates into the seed. And so then we have this theory that the blue-green algae are producing a toxin which is concentrated in the seeds, consumed by the fruit bats, and consequently eaten by the humans. And this was the theory of the mystery of they call it the Batty hypothesis. And this has been a really enduring idea for the last few decades. Once they put a ban to the consumption of fruit bats, the incidence of motor neuron disease dropped, so it's virtually zero in Guam. So this clearly implicates this source of entry for this toxin. Now, how is this relevant to modern days? Well, it turns out that blue-green algae is ubiquitous. So this is actually a picture of the Murray River earlier this year, and we can see a blue-green an algal bloom. And the Riverina area, which some of you may have had the pleasure of visiting, is rife with blue-green algae at the moment. And that blue-green algae makes this toxin, so there's potential entry into the waterways through this. And particularly if we look here at the northern Victoria, there's also hotspots of motor neuron disease. So in some regions of the Riverina, the incidence of MND is increased by 6 times. So this raises the question of whether or not there's an association with exposure to blue-green algae-derived poison. Okay, so there's some of the ideas about sporadic MND. So now I return to the inherited form of the disease. What do we know? Quite a lot. So this is a graph showing you the number of genes identified in motor neuron disease over time. And I told you before that the Australians were involved in the discovery of SOD1, and since then we've actually seen this explosion, this exponential increase in the number of genes involved. So what I can tell you right now is there are dozens of genes responsible for motor neuron disease. And we're at this point here now where I can tell you that we have solved about 70% of MND genes, and I always say that within the next 5 years or so we should crack the other 30%. And what this means is that you can give a definitive genetic diagnosis to families with motor neuron disease. But the problem with this is it doesn't help us with sporadic motor neuron disease. And remember, that was the form that was out of the blue. So this now brings me to clinical trials in motor neuron disease. So what's happening in the clinical trials space? And again, this is a very crowded space. We can see a lot of activity. If we just focus on the right side of this graph, what we do know is that clinical trials are divided into 4 stages, and the aim is to get your drug into this central area here. So we can see that there's a lot of activity in this space. These are all experimental investigational drugs. Many of them have actually failed. They've dropped out of the pipeline, and there's new contenders, and there's constant activity in this area. But to reiterate, there's been no drug approval in Australia for motor neuron disease since riluzole. Now, Australia is very active in the clinical trial space in motor neuron disease, and we have these 4 major sites. So in Victoria, we have the Bethlehem Calvary Hospital, which is engaged in clinical trials, such as the antiviral medication I spoke about. So this brings me to the exciting part of the talk. So I've given you all the background on the history of MND and the causes, and told you how miserable the clinical trial space is. So we devised a plan, and that was to establish a high throughput drug discovery program at the Florey Institute. Why did we do this? Well, we identified that there were some key issues in motor neuron disease which were impacting clinical trials. And the first problem I'll identify is that MND is not one disease. It's not a singular disease. It's highly variable from patient to patient. So when a patient arrives at clinic and they're diagnosed with MND ALS, it can then go in different directions. And what I mean by this is that there are different forms of motor neuron disease. There are forms that affect primarily the brain and the spinal cord, and these have different names. We then have different forms of the disease which can affect the facial muscles. MND has shown to overlap with a form of dementia called frontotemporal dementia, as illustrated here. So about 15% of MND patients will go on to develop frank frontotemporal dementia. Yet some patients are cognitively intact. So why is that? We have different variants that involve the extremities, such as the arm and leg, flail variants. There are different forms of motor neuron disease. We have early onset, we have late onset, we have rapidly progressive disease, but we also see cases that are slowly progressive. And I know of a case that has lingered for 35 years, and Stephen Hawking is a great example of slowly progressive disease. And then bulbar onset refers to the muscles involved in speech and swallowing. So some patients present first in this area, but the majority of patients present in the spine, so they'll present in the arm and the leg. So we have this immense diversity in the form of motor neuron disease. This is another way that we can illustrate this. So what I'm showing you here is a plot of patients over time, and this is a clinical function score. So all patients start off at about 50, and then they drop over time. So we can see that these individuals have classic motor neuron disease. They succumb to their disease within the first 27 months or so. Yet remarkably, there are patients that can survive with this disease for years. So you'll notice that you get different trajectories, and we call this a spaghetti plot. And you can appreciate from this plot that there's a lot of noise in this system, and it's no surprise that clinical trials will be doomed to fail at this rate, because these patients are not correctly subtyped. We need to accurately subtype the fast progressors from the slow progressors and enrol them in different trials. The second problem that we identified is that for the last 25 years or so, the whole scientific and research community have been focusing on this part of the pie chart. So this 10% of genetic familial motor neuron disease. And surprisingly, any drug success you find in this area will not work for the majority of patients with sporadic MND. So we have a real lack of laboratory models for sporadic motor neuron disease, and this is a major problem for clinical trials. The third problem, and I'm actually a mouse researcher, but I do appreciate this, is that we have seen perhaps an over-reliance on mouse models or animal models of motor neuron disease in the last 25 years. So what works in a mouse does not work in a human for motor neuron disease. So there's questions raised about the usefulness of mice and rats. So we embarked upon a new program which will address all of these key issues. And it's a world-first program dedicated to motor neuron disease. And I'm pleased to say it was funded last April, last year, and it was a $5 million program. and it was co-funded by some key players. So we had the Victorian State Government, Fight MND, and also the Balkan Group Proprietary Limited. And the mission of this program is very simple. What we are seeking to do is to rapidly screen clinically approved drugs, so these are prescription medications, using sporadic MND patient cells. So we're working primarily with the sporadic disease, and we're aiming to identify priority agents that we can then advance to clinical trials. And we call this drug repurposing. So we're taking existing prescription medications that you may have at home and trying to repurpose them, see if they've got any activity or potential for motor neuron disease. So this is an overview of the Fight MND drug screening program. And it begins with collecting cells from the MND patients. And these cells are then reprogrammed into stem cells, and I can take you through that. These cells are then intensely and rigorously studied and categorised. And what we do is we identify a subset of lines that we can use for drug screening. And then we can engage in high throughput drug screening. So we have a primary panel of patient cells that we use for our drug screening. And any hits, any potential drugs that we get out of this screen, we can then extend to the entire patient cohort. And then we bring this back. So this is the circular nature of this program. We bring this back to clinical trials. So how does this program work? I'm just going to take you through 2 stages of the program. So we're quite ambitious. So we're aiming to recruit about 200 MND patients through this program, and also 50 controls. And this is done in collaboration with our clinical neurologists at Calvary Healthcare Bethlehem and Barwon Health, and also through the Eye and Ear Hospital. And this is how this program works. So what we do is we can take a superficial skin biopsy from the forearm of an MND patient, and we're able to then grow those skin cells in culture. And this is a picture of these cells under the microscope, and this is a process which takes about 2 weeks. And then using molecular tricks, so using some Nobel Prize winning technology, we're then able to convert those cells back into stem cells. And this is a process which takes about 3 months. And again, these are the stem cells grown under the microscope. So we're able to turn these skin cells back in time to their stem cell state. And then that gives us the power to then turn them into motor neurons. So we can turn them into any cell type we desire, but in this case, we're interested in motor neurons. And this is a process that can take at least a month. And shown here are some images of these cells. And what we're trying to achieve here is MND in a dish, and it's personalised MND in a dish, if you think about 200 patients that we're recruiting. So stage 2 is where we get into our drug screen. So let's imagine that we now have an infinite resource of motor neurons, the affected cell type from patients living with motor neuron disease. What we're able to do is then drop them into these assay plates, about 10,000 cells per well. And imagine that we have 20 or so of these plates, and we can then subject them to what we call high throughput drug screening. And this is facilitated by robots. I'm going to talk here a little bit about some of the robots that we've installed in the Institute. So what you're looking at here is a $1 million machine or a robot that was installed. And it's capable of taking these so-called drug libraries, which contain thousands or tens of thousands of drugs, and then treating these motor neurons. The second phase of this is that we have this is the Australian first version of this microscope. So this is a high-powered, high-resolution microscope which can then track the health and survival of these motor neurons in response to drug treatment. And the ideal hope of this program is that we're able to generate drug hits that we can then take forward for further validation. I just wanted to point out that using conventional manual techniques, we've estimated that we can increase the efficiency of drug screening by about 150 times using this approach. I wanted to show you a video that we've generated. So now you're looking down into one of these wells in these assay plates. And what we've done is we've labelled the motor neurons in green, so we can make them glow green so we can identify them. And what you can see here is movement over time. If you track this motor neuron here, you can see that these cells are quite mobile. They put out these connections and they're sensing their environment. And this is the key parameter or readout that we're looking for. So here we have a motor neuron, coloured in green, And if we track this cell over time, you can see that that green is eventually extinguished and that cell dies. And that's the key measure that we're looking for. We're looking for survival of these cells under the microscope. So here's another way that we can picture this. We're modelling motor neuron disease in the dish, and we're looking for a loss of that green signal over time as this cell converts into a sick, diseased motor neuron. And what we do is we can score living cells versus dead cells, and we can generate these plots. So this is not too dissimilar from that survival graph that I showed you before for riluzole treatment in motor neuron disease patients. And what we have here is each little increment on this graph represents the loss of a motor neuron over time. And this is a theoretical plot, and we're looking here at the health of a control motor neuron, and we would estimate that they can survive up to a week. But a motor neuron that's affected by MND has a reduced survival, and that's measurable by this method. So we can now see a shift towards the left of this graph. What we now do is we intervene. So once we establish a drug hit, so to say drug X, we're looking here to bring that motor neuron back to life to promote its survival. So now we're talking here about clinical trial in a dish. So here we have a motor neuron analysis. This is a motor neuron affected by MND. And using drug X, we're able to bring back the survival. But the real power of this high throughput system is we can now consider combinations of drugs targeting different aspects of the disease. And here we have, now we have an additive effect. So we're looking now at drug combinations. And again, this is a nice correlate of a clinical trial. So think of this as a clinical trial in the dish, but much more efficient. So here we have a local who dropped in to donate his skin cells. So this is Neil Danaher. He was the first patient to donate to our program. So we're very lucky to have him in our program. And here are some action shots from the lab collected. So this is Chris, and we also have Catherine over here who are running the program. And you can see that they're at different stages of the program. So here we have the robot which plates out the drugs. We've got some motor neurons, got some analysis here, and eventually a lot of microscope use. So I just wanted to give you an update on progress on this program. And I'm very pleased to tell you that we started patient recruitment last December, and we were aiming to recruit 20 patients per month. And we're on track. So we're aiming to get ideally to 200 patients, but right now I can tell you that we've steadily increased our patient number to about 150. So we're certainly on track to hit our aim of 200 patients. And then I just wanted to break down those 150 patients. So what I can tell you is that this is primarily a Victorian initiative. So we've enrolled 120 patients from Victoria, but we have taken some interstate patients. We've even had patients fly in from Perth or Hobart because they're so keen to be part of this initiative. And then these are just some of the characteristics of the patients. So we've currently enrolled predominantly male patients. So this disease is actually 50% more common in men than women. So this fits. And then age of onset. So the majority of the patients, MND is usually a late onset disease, so it spikes usually in the 50s to 70s. So we're capturing that patient cohort, but it's not unusual to capture patients with young onset motor neuron disease. And this is precisely what we want to do. We want to capture the entire clinical spectrum of disease, so we can model this. And then I told you before that MND, diagnostically, there are different forms and different variants. So majority of the patients we're capturing are classical ALS, but we are enrolling other forms of the disease, the slowly progressive form, the brain form, the spinal form. And then sort of onset, so these patients present in different parts of their anatomy. So the majority of our patients have spinal onset where they present in the leg or the arm, but we do have patients presenting in the muscles of speech and swallowing, and also generalised. So again, capturing that whole clinical picture. And then progression rate, we've just estimated here. We've got the slow progressors, the rapid progressors, and then the classic. We're also capturing some of the genetic forms of motor neuron disease, so that these are good, solid, positive controls to have. And we do have, you may recall that first gene that was identified 25 years ago. So we have those patients represented within our cohort amongst other different forms. So it's a very exciting program. Recruitment is certainly well underway. Drug screening will be underway in the second half of this year. We have our first drug libraries. We're very excited to start generating hits, but I just wanted to end my talk by telling you that I think there is much more scope and potential to this program. So we think of this as a unique resource for Australia. It's certainly the largest collection of stem cells from motor neuron disease patients, and there are so many other questions that you can address. And there are different directions that we're taking this program in. So one of our interests at the moment is to really profile these motor neurons and really get into the genes and the molecules behind the disease. And what we would like to do is actually subtype or subgroup patients at the genetic and molecular level using profiling technology. And this is called precision medicine. So this is a term which is widely, it's actually widely practiced in oncology and hematology, but it's in its infancy in neurology. And what it means is it's really about bringing the drug back to the cause of that individual's motor neuron disease. So this is one particular area that we're hoping to branch off to. We also want to build more complex models. So there's these so-called brain in a dish systems where you can build in complexity and you can start to model those brain structures that I showed you that Campbell drew over 100 years ago in a dish. And also brain on a chip is also another approach where you can interface these cells with technology in order to deliver a blood supply. And you can grow them on a chip. So I just wanted to end by saying this is the lab. We've donned our beanies for the big freeze, which is next week. So I just wanted to point out, how can you help our research continue? Well, there's obviously two ways. One is you can buy a beanie, so they're available at reception. I've got a bag behind me to support Fight MND, but also donate to the Florey Institute so we can keep this program going, we can continue our recruitment of patients, and keep the drug screening alive. So thanks very much.